Our next talk is by Misty Riddle from the Department of Molecular, Cellular, and Developmental Biology, Reprogramming Cell Fate and Remodeling Organs in a Tiny Worm.
This is Matthew. He needs a kidney. But donor organs are scarce, and even more scarce for Matthew because he has a rare blood type. If he does find a suitable donor kidney, there's still a chance that his body will recognize the organ as foreign and reject it. But Matthew, like all of us, is made up of trillions of cells. And although these cells have very different sizes, shapes, and functions, they all contain identical genes. This means that each one of Matthew's cells has the instructions to build a kidney. So theoretically, it's possible to use Matthew's own cells to build him a perfectly matched kidney. The problem is, once a cell adopts a particular identity, it's very resistant to changing. My name's Misty, and I've spent the last 4 years figuring out how to coax stubborn cells into changing their identities. And what I want to show you now is I've been successful in reprogramming cells and remodeling organs in a simple worm. These are two of the adult worms that I'm talking about. They're transparent, so we can see all of their internal organs. And they're incredible models for studying cell reprogramming for 3 reasons. 1, they're simple. We have trillions of cells. They have exactly 959 cells. 2, we have an incredible amount of information about them. For example, this was the first animal to have its entire genome sequenced. And 3, importantly, they're similar enough to us that at least the basis of what we learn can inform human medicine. One of the incredible tools that we have for working with them is the ability to fluorescently label specific cells and organs. Here's the same worms illuminated with a UV light source. We've labeled the intestine with blue fluorescence and the uterus in the mid-body of the worm and pharynx, a heart-like organ in the head region, with green fluorescence. Here those 2 organs are shown more closely. I found that using a single protein, I can access the instructions for building an intestine in the pharynx and the uterus and essentially reprogram and remodel these organs to intestine. Here you see blue fluorescent intestine where the uterus and green pharynx used to be. And remember, we're doing this in a live worm that we have all this information about, so it really allows us to really figure out how exactly this conversion is taking place. Matthew has done an incredible job at raising awareness for organ donation. As you can see, he got way more than 1 million likes on Facebook. And I'm not going to claim that from what I've showed you today, we understand how to build him a kidney. However, it does give us a really valuable tool to be able to understand the ins and outs of cell and organ remodeling. And hopefully with that information, we can one day overcome the challenges of organ transplant to use a patient's own cells to build them perfectly matched organs. Thank you.